Hello friends, myself Dr. Akhilesh. In this video, I will discuss the important MCQs on cell membrane. So this topic is also important for the NEET, NEXT, INI-CET, FMGE, USMLE, PGI, PGI and various other entrance exams. So let us look at the questions here. The first question is, the plasma membrane is mainly composed of. So to solve this question, you need to remember the composition of the cell membrane. So what is the composition of the cell membrane? So this plasma membrane, you can see here, it is made up of, number one thing is, it is made up of the proteins. Number two thing, it is made up of lipids, and the main lipid is the phospholipid. The number 3 is the carbohydrates. And so proteins, lipids, carbohydrates, and in this you can see here the phospholipid, and in the phospholipid you can see, so this round structure, this is called as the head of this phospholipid, which is made up of the phosphate. So this head is made up of the phosphate group, and these are the tail of this phospholipid. So it will constitute the major lipid in the cell membrane, and you can see here that, so this phospholipid, it has the two layers. So this cell membrane is made up of lipid bilayer, and in this lipid bilayer you can see here that the central part of this lipid, so central part of this lipid is hydrophobic. Hydrophobic means it will repel the water. So it is hydrophobic, so it will repel the water. And the peripheral part of this phospholipid, the peripheral part of this phospholipid, which is made up of the phosphate group, so this peripheral part is hydrophilic, and this is the water loving. So this phospholipid central part is hydrophobic and the peripheral part is hydrophilic. So this cell membrane is made up of two layers of the phospholipid. So therefore we also say that the cell membrane is lipid bilayer. So lipid bilayer, and these phospholipids, they are responsible for the fluidity of the cell membrane. So it is responsible for the fluidity of the cell membrane. Right. And in the proteins you can see here, the proteins, they are of two types. So one is called as the integral proteins and other are called as the peripheral proteins. So two types of proteins are there. In the integral protein is also called as intrinsic protein. It is also called as the intrinsic proteins, and the peripheral proteins, they are also called as the extrinsic proteins. So two types of proteins are there, integral and peripheral proteins. The integral proteins, you can see here some integral proteins, they are just embedded in this lipid bilayer, and some integral proteins you can see here, they pass through this cell membrane. So such proteins, integral proteins which pass through the membrane through and through, so they are called as transmembrane proteins. Right. So third component is the carbohydrate. So therefore the approximate composition of the cell membrane is the proteins, which constitute around 55% of the cell membrane, the phospholipid 25%, cholesterol 13%, other lipids which include the lecithin also, so it will include the 4%. So this phospholipid, cholesterol and other lipids, so they will constitute approximately 42% of the cell membrane. The carbohydrates, they will constitute 3% of the cell membrane. So therefore the percentage of protein is more than the lipid and more than the carbohydrate. So this percentage is in terms of the area covered in the cell membrane. So of all the given components, which is the main main constituent of cell membrane. So you can see here the main constituent of the cell membrane will be the proteins, that in terms of the percentage of the area covered on the cell membrane. But in terms of the masses, the protein and lipid compounds, they are in the ratio of 50-50 or 1:1. This means that in terms of the masses or dry mass, the protein compounds and lipid compounds, they are in the ratio, equal ratio, that is 1:1 or 50-50. So therefore, look at the question here. The plasma membrane is mainly composed of, so in terms of the percentage, so we can say the cholesterol is 13%, carbohydrate is 3%, phospholipid 25%, proteins they are 55%. So out of these, the protein, they are the main or major component of this cell membrane in terms of the area occupied in the cell membrane. Now let us look at the second question here. So on weight basis, the membrane contains protein and lipid in the ratio of, so this I told you that in terms of the masses, the cell membrane contains protein and lipid compound in equal ratio, that is 1:1. So protein compounds and lipid compounds, in terms of dry masses, protein compounds and lipid compounds, they are in equal ratio 1:1 or 50:50. So our answer will be what? So our answer will be 1:1, or you can say that 50-50 or 1:1. So answer will be here B, 1:1. Now let us look at another question here. So another question is, which important component of the cell membrane has carbohydrate moiety? Carbohydrate moiety means the structures present in the cell membrane which have the carbohydrate compound. The options you can see here: the gangliosides, triacylglycerol, sphingomyelin, cerebrosides and phosphoglycerates. So you can see here 1, 2, 3, 4, 5 options are there. So 5 option questions are usually asked in the PGI exam, and they may have more than one correct answer. So they may have more than one correct answer. So to answer them, we should have the thorough knowledge also. So more than one correct answer, remember this. So they may have the multiple answers also. So let us see which of these options they have the carbohydrate compounds or carbohydrate containing compounds. So carbohydrate containing compounds, one are the glycosphingolipids. So glycosphingolipids, they are the carbohydrates that contain lipid, and they also have the ceramide as the backbone. An example of such glycosphingolipids that include the galactosyl ceramide, glucosyl ceramide, which is also called as cerebrosides, and gangliosides. So galactosyl ceramide, glucosyl ceramide or cerebrosides, gangliosides, they are some examples of the carbohydrate moieties. So let us look at the option here again. So gangliosides, they are the carbohydrate moieties, they contain the carbohydrate. The triacylglycerol, so this is the fatty acid plus the alcohol. So alcohol here is the glycerol, fatty acid plus glycerol, so this is a lipid, right? Sphingomyelin, again it is a lipid. The cerebroside, they again contain the carbohydrate compounds. The phosphoglycerate, so phosphoglycerate means phosphate plus glycerol, so they are also not the carbohydrate moieties. So here the correct answers are A, gangliosides and ceramides. So you can see here that the carbohydrate moieties are galactosyl ceramide, glucosyl ceramide, cerebrosides and gangliosides. So these options A and D are correct here as the carbohydrate moiety. Now let us look at the other question here. Another question is, eukaryotic plasma membrane is made up of all except. So which is usually not present in the eukaryotic cell membrane. So carbohydrate, they are present here, and I have shown you here the approximate composition of the cell membrane. So protein, they are present, phospholipid, cholesterol, other lipids including the lecithin and carbohydrate. So this is the approximate composition of the cell membrane. So which one is not present in the eukaryotic cell membrane is they are the triglycerides. So these triglycerides, they are usually not present in the cell membrane, eukaryotic cell membrane. So in this option, so made up of all except, so our answer will be which one? Triglycerides. So carbohydrate, they are present, right? Cholesterol, it is also present. This lecithin, which is also lipid, it is also present in the cell membrane. But these triglycerides, they are usually not present in this eukaryotic cell membrane. So answer will be the B, triglycerides. Then let us look at the another question here. The fluidity of the plasma membrane is increased by. Now this question is asking about the fluidity. So to understand the concept of the fluidity, you should have the knowledge of this phospholipids. So to answer this question, we should have the knowledge of the phospholipids because they will affect the fluidity of the cell membrane. So what is the structure of this phospholipid? You can see here. So this is the structure of the phospholipid. So this phospholipid, they have one head which is made up of the phosphate, and this head is hydrophilic. Hydrophilic means water loving. And this is the tail. This tail is made up of the lipid. The lipid is made up of what? Fatty acids plus alcohol. So these fatty acids, they can be. So one type of fatty acid is the saturated fatty acids. Saturated fatty acids, and other can be the unsaturated fatty acids. Unsaturated fatty acids. So saturated fatty acids, for example, stearic acid, palmitic acid. So what these saturated fatty acids they do, that when they are present in the lipid tail, so the lipid tail will be straight and parallel tail, right? So if the tail consists of saturated fatty acids, so example of saturated fatty acid I just mentioned is stearic acid and palmitic acid. So if tail consists of saturated fatty acid, the tail will be straight and parallel tail. So these straight and parallel tails, they favor tight packing in the cell membrane, and they will increase the solidity of the cell membrane, and when solidity will increase, the fluidity of the cell membrane will decrease. So remember this, that the saturated fatty acids, remember this, S for saturated fatty acid, they will increase the solidity. So S for saturated fatty acid, S for solidity. So they will increase the solidity of the cell membrane, and when the solidity will increase, what will happen to the fluidity? So fluidity will decrease. So saturated fatty acids, they will decrease the fluidity. And remember here the example of the saturated fatty acid. So one example is stearic acid. So this also begins with S. So stearic acid, it will also begin with the word S. So S for saturated, S will increase solidity. S example is stearic acid. So saturated fatty acid, they will decrease the fluidity of the cell membrane. But if the lipid tail consists of unsaturated fatty acids, so examples of unsaturated fatty acid is linoleic acid, oleic acid, and linolenic acid. So you can remember the mnemonic here, LOL, LOL. So unsaturated fatty acids example, linoleic acid, oleic acid, and linolenic acid. So when they are present in the lipid tail, the lipid tail will be unparalleled, and there will be bending in the lipid tail. The bending is also called as kinked or kinking of the tail. So this unparalleled and kinked tail, they will favor the loose packing of the cell membrane, and the packing is loose, it will decrease the solidity of the cell membrane, and it will increase the fluidity of the cell membrane. So unsaturated fatty acid, what they are doing, they are increasing the fluidity of the cell membrane. So they are increasing the fluidity of cell membrane. So remember LOL, LOL, linoleic, oleic and linoleic acid, they will increase fluidity of the cell membrane. And you have to also remember that this oleic acid is the most abundant unsaturated fatty acid in the eukaryotic cell membrane. It is most abundant unsaturated fatty acid in eukaryotic cell membrane. Oleic acid is the most abundant unsaturated fatty acid in eukaryotic cell membrane. So now let us look at the options here. The fluidity of this plasma membrane is increased by, so stearic acid, palmitic acid, they are the saturated fatty acid. So they are the saturated fatty acids, and they will increase the solidity. They will increase the solidity of cell membrane, and they will decrease the fluidity of the cell membrane. The linoleic acid, oleic acid and the linolenic acid, they are the unsaturated fatty acids, and they will increase the fluidity of the cell membrane. So answer here will be the linoleic acid. So remember here, the linoleic acid, oleic acid and linolenic acid, they are unsaturated fatty acids, and they will increase the fluidity of the cell membrane. Now let us look at another question here. Addition of PUFA. PUFA is polyunsaturated fatty acids in the cell membrane. They will cause what? So what they will cause? So PUFA is polyunsaturated fatty acid. So what are polyunsaturated fatty acids? So polyunsaturated fatty acids example are linoleic acid, linolenic acid, arachidonic acid, and the example of monounsaturated fatty acids are oleic acids. So these unsaturated fatty acids, just I told you that these unsaturated fatty acids, what they will do is they will increase the fluidity of the cell membrane. So let us look at the options here. So addition of the PUFA in the plasma membrane, they will increase the fluidity of the cell membrane. So answer will be the B here. Now let us look at another question here. The another question is the transition temperature of the lipid bilayer of the cell membrane is increased by. Who will increase the transition temperature? So here you have to remember what is the transition temperature, what is meant by transition temperature of the cell membrane. So this transition temperature of the cell membrane is also short form is TM is used here, transition temperature. So the word is transition and temperature. Two words are there, transition and temperature. So the transition temperature is that temperature above which the ordered arrangement of the cell membrane is converted to disordered arrangement. So ordered arrangement is converted to disordered arrangement means simply you can also understand. So it will, the transition temperature is the temperature above which. So ordered arrangement is present in where? Solid, right? And converted to disordered arrangement is present into where? So liquid. So temperature above which the solid become liquid, or you can also say that the temperature above which this ordered arrangement is converted to disordered arrangement, that is called as a transition temperature. So if there is more solidity, if the membrane is more solid, so what will happen here? If the membrane is more solid, then more temperature is required to convert into liquid. So more solid, more temperature required, so it will have the more transition temperature, or it will have high Tm. So you can also say that high Tm. So more solidity will have the high Tm or more Tm. The saturated fatty acids, this I told you, example stearic acid, palmitic acid. So saturated fatty acids, stearic acid and palmitic acid, what they do? They will increase the solidity of the cell membrane, and when solidity of cell membrane will increase, then what will happen to the transition temperature? So more Tm, more temperature required to convert the order arrangement into the disorder. So it will increase the Tm. And just I told you the solidity will decrease the fluidity. So remember this, the saturated fatty acids, they will do two things. So saturated fatty acids, they are doing two things. Number one is they are increasing the solidity of the cell membrane, and when the solidity will increase, so what will happen when the solidity will increase? So it will increase the transition temperature also. And when the solidity will increase, what will happen to the fluidity? So this fluidity of cell membrane will decrease. And what about the unsaturated fatty acids? So this unsaturated fatty acids, for example, I just told you LOL. L stands for linoleic acid. O stands for oleic acid. L stands for linolenic acid. So unsaturated fatty acids, they will decrease the solidity of the cell membrane, and when solidity will decrease, so what will happen to the transition temperature? So transition temperature of cell membrane will also decrease, and it will increase the fluidity of the cell membrane. So the question is that the transition temperature of cell membrane is increased by, so who will increase the transition temperature? So transition temperature will be increased by the saturated fatty acids. So answer here is the B, saturated fatty acids. Right. So example of saturated fatty acids is stearic acid, palmitic acid. They are just few examples of the saturated fatty acids. Now let us look at another question here. The transmembrane region of protein is likely to have. So now this you have to remember here, the transmembrane region of the protein in the membrane. So you have to remember here about the protein in the cell membrane. So this protein in the cell membrane, you can see the diagram again. The protein in the cell membrane, they are of two types. One is the integral protein, or also called as the intrinsic proteins, and number two is the peripheral proteins, also called as the extrinsic proteins. So two types of the proteins are there, and the integral proteins which are embedded in the cell membrane, or which are the transmembrane proteins. So this in the integral protein, the central part of this protein. So this protein, central part of the protein which is embedded with the lipid of this phospholipid, so they contain the hydrophobic amino acid. So it is made up of the hydrophobic amino acids. So this all the central part or this transmembrane part of this transmembrane proteins, they contain hydrophobic amino acids. And the peripheral part of these proteins, so they contain the hydrophilic. So they contain the hydrophilic amino acids. Right. So this portion, the peripheral part will contain the hydrophilic amino acids, and this central portion in the protein, they will contain the hydrophobic amino acids. This arrangement is very similar to the phospholipid, where the central part of this phospholipid is hydrophobic and the peripheral part of this phospholipid is hydrophilic. So this arrangement is very similar to that of the phospholipid. So in the phospholipid as well as in the protein, the central part contain the hydrophobic area and the peripheral part they contain the hydrophilic area. So now let us look at this question again. The transmembrane region is which is central part of this the cell membrane. The transmembrane region the protein is likely to have. So they have the hydrophobic amino acids. So they will have the hydrophobic amino acids. So answer here is this, the B is such of hydrophobic amino acids. Now let us look at another sample question here. The integral proteins of the plasma membrane, they act as. So now you have to know the functions of these proteins in the plasma membrane. So in this question it is asking the integral proteins. So let us look at what are the functions of the proteins in the cell membrane. So you can see here again that I told you that the proteins are of two types, integral proteins and the peripheral proteins. So two types of proteins are there. So what is the function of this integral protein and what are the functions of these peripheral proteins? So in the diagram you can see here one function is they act as channel. So integral proteins, they can act as channels or pores, they act as carrier proteins, pumps, enzymes, receptors, adhesion molecules, and they also participate in the intracellular signaling. So these are the various functions of the integral protein. And what are the functions of these peripheral proteins? The peripheral proteins, they also act as enzyme, the controller of transport of substances through the cell membrane pores. They also participate in intracellular signaling and they can form a submembranous cytoskeleton, as in example is the ankyrin and spectrin. So these are some functions of the peripheral proteins. So let us look at the option again. So integral proteins, here it is asking about integral protein. They act as pumps, yes they will act as pump. Ion channel, yes they will act as ion channel. Pores, yes they will act as pores also. So here answer, all the A, B, C options are correct. So here will be the all of the above are correct. So correct answer here is the D. Now let us look at another question here. Which of the following membrane has highest protein content per gram tissue? So now here in the previous question I told you that the protein compound and the lipid compound, they are in the ratio of 1:1 in the cell membrane. The protein and lipid compound ratio is 1:1. But in this question it is asking that which has the highest protein content per gram tissue. So this means that there is variation in the various membrane where the protein and lipid ratio, it can be altered than 1:1. So let us look at here table here. So let us look at the table. So membrane and the protein is lipid ratio. In the inner mitochondrial membrane, the protein to lipid ratio is 3.2:1. Right, here protein is in very high content. So 3.2 times protein as compared to the lipid. In the sarcoplasmic reticulum, so this ratio is 2:1. So this protein to lipid ratio is 2:1. In the outer mitochondrial membrane and the RBC membrane, this ratio is 1.1:1. So these are the three examples of the membranes where they have the high protein content. But in the myelin sheath, so this protein is to lipid ratio is 0.23:1. So this myelin sheath has the more lipid than the protein. So myelin sheath is basically made up of lipid, layer of the lipid, then there is layer of the protein, and then again there is layer of the lipid. So it has the two layer of the lipid in which the protein layer is sandwiched. So in the myelin sheath, the lipid is more than this proteins. So this ratio is 0.23:1. The protein lipid ratio is 0.23:1. So this question is asking about which of the following membrane has the highest protein content per gram tissue. So in the inner mitochondrial membrane, this ratio is 3.2:1. Outer mitochondrial membrane, this ratio is 1.1:1. In the plasma membrane, the ratio is 1:1. As I told you previously, myelin sheath, this ratio is 0.23:1. So which one has the highest protein content per gram tissue? So answer will be the A, inner mitochondrial membrane. Now let us look at another question here. The protein is to lipid ratio in the myelin sheath is. So just I told you that this ratio is in the myelin sheath, the protein to lipid ratio is how much? 0.23, and this is the minimum ratio where the protein content is very less. So myelin sheath, so this ratio will be 0.23:1. So answer will be the D here. Let us look at the another question here is, in the cell membrane, the following statement is or are true. So again here 1, 2, 3, 4, 5 options are there. So this question has been asked in the PGI exam, and remember here, so it can have multiple correct answers. So it has multiple correct answers. It means it may have more than one correct answer. So remember this, in the PGI exam, the questions are framed in such a way that they may have more than one correct answer. So we have to be thorough knowledge of each options. So let us, what are the various options here? The option A, lipids are regularly arranged, lipids are symmetrically arranged, protein exhibit lateral displacement, membrane is dynamic structure, choline containing phospholipids are located mainly in the inner layer. So let us discuss this each option one by one. So option A is lipids are regularly arranged, B is lipids are symmetrically arranged. So regarding this A and B, remember here is the membranes are asymmetric structure, they are not symmetrical. Remember, they are asymmetric structures. Number one thing, to remember this, the membrane are asymmetric structure. The protein, lipid and other components, they are not regularly arranged. So the option A and B are wrong, which was given there. And remember here, they are not regularly arranged and they are not symmetrically arranged also. So they are not symmetrically arranged also. Right. Then let us look at the option C. Option C is proteins exhibit lateral displacement. Yes, the proteins exhibit lateral displacement in the form of the diffusion in the plane of their membrane. So this option C is correct. Option D. The option D is membrane is dynamic structure. So regarding this option D, yes, the membrane is dynamic structure, right? The lipid, the various integral proteins, they move from one point to another point within the plane of the membrane. That is also called as lateral diffusion. So this option D is the correct one. That is the membrane is dynamic structure. Regarding the option E, the option E is the choline containing phospholipids are located mainly in the inner layer. It is saying the inner layer. So this option is wrong because the choline containing compounds, they are located mainly in the outer layer, not in the inner layer. So this option E is also wrong. So choline containing phospholipid, they are located mainly in the outer layer, whereas the aminophospholipids, they are located in the inner layer. Aminophospholipids are located in the inner layer. So in this question, option A, lipids are regularly arranged, is wrong. They are not regularly arranged. Lipids are symmetrical structure, they are again wrong. Symmetrically arranged, they are wrong because they are asymmetrically arranged, right? Option C, protein exhibit lateral displacement, yes, it is correct. Membrane is dynamic structure, yes, it is also correct. And the choline containing phospholipids are located in the inner layer, this is wrong because this choline containing phospholipids are located in the outer layer. So option C and D, these two options are correct in this question. Now let us look at another question here. True about lipid bilayer of the cell wall. So lipid bilayer cell wall. Again here 1, 2, 3, 4, 5 options are there, and this question appeared in the PGI exam, and this may have more than one correct answer. Remember this. So what is the option A? Asymmetrical arrangement of the cell membrane component. B. Lateral diffusion of the ions. C. Symmetrical arrangement of cell membrane components. So A and C are just opposite statement. Not made up of amphipathic lipid. Membrane repair themselves by rapid resealing. So let us look at these options one by one. So option number A, asymmetrical arrangement, and option number C, symmetrical arrangement. So out of this A and C, so regarding this option A and C, just I have told you in the previous question that the cell membrane components are asymmetrically arranged. So option A is correct and C is wrong. And I have told you that the protein and lipid and other components of cell membrane are not regularly and symmetrically arranged. Means they are not regularly arranged and they are not symmetrically arranged. So option C is wrong here, A is correct. Option B, there is lateral diffusion of the ions. So ions, they do not diffuse laterally, they will diffuse across the membrane, right? So they will diffuse the membrane across the movement. They will cross this membrane through and through, not this lateral displacement. So this lateral diffusion is seen in the case of the proteins, right? And they will, in the case of this, the lipids, they will move in the lateral direction. But this ions, they will move across the movement, across the cell membrane, through and through the cell membrane. So this option B is also wrong. Option number D, not made up of amphipathic lipid. Amphipathic means which has both the hydrophilic and hydrophobic ends. So I have just told you that the cell membrane is made up of what? The cell membrane is made up of lipid bilayer. So this is the head part of the phospholipid. This head is made up of which group? The phosphate group, and this is what? So this is the hydrophilic. The central part, the tail, it is made up of the lipid, and this lipid is hydrophobic. So it is the hydrophobic. So therefore this lipid bilayer has hydrophobic as well as the head, which is made up of phosphate, it has the hydrophilic, and the tail has the hydrophobic. So this, the cell membrane, the phospholipid bilayer has both hydrophilic and hydrophobic. So this is the amphipathic. So this is the amphipathic. So the option D, which is mentioning that not made up of amphipathic. So this is the wrong statement, because the cell membrane is made up of amphipathic lipid, means it has both hydrophilic and hydrophobic end. Option E. So option E is that the membrane repair themselves by rapid resealing. Yes, it is true here, because after an injury, the cell membrane, they repair themselves by the rapid resealing. They will reseal themselves very rapidly. So this option E is correct here. So which statements are correct in this question? So A, asymmetrical arrangement, this is correct. Lateral diffusion of ions, no, it is the across movement. Symmetrical arrangement cell membrane component, no, it is wrong. Not made up of amphipathic, it is wrong, because it is made up of amphipathic lipid. And the membrane repair themselves by rapid resealing, this is the correct one. So this option number A and option E, they are correct in this question regarding this lipid bilayer of the cell wall. Now let us look at another question here. A cell membrane is damaged by insertion of microneedle. So this repair shall occur by which of the following process? So it is asking that if the cell membrane is damaged by a microneedle, so it will repair by which process? So to answer this question, we need the following concepts. Number one is, whenever there is injury to the cell membrane, So how this cell membrane they repair themselves? One way is that the fusion of these vesicles. Vesicles are made up of phospholipid. So these phospholipids they will fuse at the injury site. The vesicle will fuse at the injury site. Number one. Number two, then the cell membrane will constrict around the area of the damage or disruption. The second method. The third one is the endocytic mechanism. In case resealing of larger membrane disruptions. If the membrane damage is larger, then there will be endocytic mechanisms where again the vesicles will fuse with here with the cell membrane. Number four is the pinching of the membrane at the injured site and shedding the injured membrane into the extracellular space. Right. So smaller disruptions are there, so they will pinch off and shed off the injured membrane. The remaining area it will reseal themselves. And the number 5 is the hydrophobic interaction between the lipid. So these are the various 5 methods. One is the fusion of the vesicle at the injury site. Number 2 is constriction of the membrane. Number 3 is endocytic mechanisms where the vesicles will fuse here. Number 4 is pinching and shedding of the injured area. Number 5 is the hydrophobic interaction between the lipids. So this repair mechanism, the membrane will undergo which repair mechanism? So that will depend upon the size of the injury, right? But whatever is there, so this membrane will rapidly reseal the membrane which is damaged. So it will, remember here, rapidly reseal. And if the damage is very small, right, less than a nanometer, so they will repair themselves by hydrophobic interaction between lipids. So they will repair by hydrophobic interaction between the lipids if the damage is less than nanometer, right? But if the damage is more than few nanometers, right, if the more than few nanometers, then they require active membrane repair mechanisms. For example, if the repair is very large, that is hundreds to thousands of nanometer, so they will require what? Calcium dependent exocytosis and endocytosis, which will involve the fusion of the vesicle with plasma membrane to patch the compromised membrane, and they will do so by rapidly resealing. So they will do so by rapidly resealing here, right? So this exocytosis, they will facilitate the resealing of the lipid by layer. So in this question, it is asked that insertion of a micro needle. So micro needle is 1000 times more than the nanometer, right? So micro is 1000 times more than this nano. So if this injury is 1000 times more than the nanometer, so how it will repair here? So it will repair here by rapid resealing. So it will repair by rapid resealing. Remember this word, it will rapid resealing there. It will rapidly reseal the lipid by layer, right? So lipid by layer by this endocytic or exocytic vesicles, endocytic or exocytic vesicles. This hydrophobic interaction is for if the injury is nanometer, right? So nanometer size injury. So here it will heal by itself by hydrophobic interaction. But in this question, it is asking the micro needle is there. So remember here it will heal by endocytic and exocytic vesicles, and it will rapidly reseal with this lipid bilayer. Answer will be the B here. Now the another question is, which of the following is seen in association with membrane raft? Now this you should have the knowledge of what is the membrane raft and what does this membrane raft contain. So membrane raft is that area of the cell membrane which is enriched in. So it is enriched in sphingolipids, for example sphingomyelin. It is enriched in glycosphingolipids, for example GM gangliosides. It is also enriched in saturated phospholipids, cholesterol, and they also contain certain GPI-linked proteins. So these 1, 2, 3, 4, 5. So membrane lipids rafts are enriched in these 5 different components. Right. So now let us look at the options here. What are the options here? Which are the following seen in association with membrane raft? So in this, out of these 5, sphingolipid, glycosphingolipid, saturated phospholipid, cholesterol, GPI linked proteins, which one is matching here? So this one is matching here, that is the GPI anchored proteins. So these membrane rafts, they are enriched in sphingolipid, glycosphingolipid, saturated phospholipid, cholesterol, and GPI linked proteins. So remember in this question here. Now let us look at the another question here. Another question is structure without membrane. So which of the following do not have the membrane? So Golgi apparatus, it has the membrane. So membrane is present in the Golgi apparatus. In the sarcoplasmic reticulum, again the membrane is present. In the lysosome also they have the membrane, but the nucleolus they do not have membrane, or they does not have, so they does not have membrane. So which structure is without membrane? So answer will be the nucleolus, though this nucleolus do not have the membrane. The next question is that the plasma membrane markers is या are. So this type of question they are very frequently asked in the AIMS exam or this INICET exam. So these types of questions are very frequently asked in this exam here. So you have to remember the plasma membrane markers. What are the markers of the plasma membrane? So these all are the enzymes. So which enzymes are the markers for this plasma membrane? So the plasma membrane markers they are, one is the sodium potassium ATPase, second is the 5' nucleotidase, the third one is adenylyl cyclase. So sodium potassium ATPase, 5' nucleotidase, adenylyl cyclase, they are the markers of the plasma membrane. So in the question, which are the correct one? So this question A, B, C, D, E, 5 options are there. So this is again the PGI question, and this PGI question has more than one correct answer here. So the correct answers are 5' nucleotidase, adenylyl cyclase, and this sodium potassium ATPase. So these 3 are the markers of this plasma membrane. So answer A, answer D, and answer E, they are correct here. So these are the plasma membrane markers. So if you want to go through more MCQs in the physiology, you can visit the Facebook page Physiology MCQ, and you can also go through this book, the Conceptual Physiology Review, which is available on the Amazon. You can buy from the Amazon. So if you like this video, then you can subscribe the video and you can press the like button in this video. Thank you very much for watching this video. So thank you very much.